So, in this lecture, we're going to be covering muscular physiology. There's going to be three types of muscle tissue we'll talk about. There's skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is under voluntary control. Let me get some pointers out here. Our skeletal muscle is under voluntary control. Your cardiac muscle and smooth muscle are under involuntary control. Therefore, skeletal muscle I can think of and I can say I want to contract this muscle and I can have a desired action. With cardiac muscle, I'm only going to find that in the myocardium of my heart. I'm only going to find that in the myocardium of my heart, and I cannot tell my heart I want you to beat faster or beat slower. It's just not possible. But you could do activities to cause your heart rate to raise. You just can't think of it and change it on your own. Or tell your heart you want it to stop beating for 5 seconds or something like that. And then the next one is smooth muscle. Smooth muscle you'll find lining all of your hollow organs. And smooth muscle will be there to help with movement. Once again, we have no control over the smooth muscle. Now your cardiac muscle and your skeletal muscle will have something in common because these two types of muscles are what is called striated. We'll talk about that in the future. And smooth muscle is not striated. So, skeletal is voluntary, cardiac and smooth are involuntary. Skeletal muscle and cardiac muscle are both striated. Skeletal muscle is the muscles that's going to help us move our bones about the joints. Cardiac muscle is only found in the myocardium, the heart wall of the heart. Therefore, it's not found in blood vessels or the blood vessels of the heart. And smooth muscle will be found in all of our hollow organs, like blood vessels, esophagus, stomach, small intestines, all that stuff. So with muscle, it's going to have some special characteristics. The 4 big ones are going to be it is excitable. Therefore, if it receives some sort of electrical input or stimulus, it will respond. Its typical response will be contraction, which is our next part, which is contractibility, or contractility, which means the muscle will shorten. And then there's extensibility, right? So before you or after you exercise, people like to stretch out their muscles. So literally they stretch out like rubber bands, and when you release the pressure, they recoil. So they also will have an elastic type of property. The functions are going to be for movement of our bones and our fluids. So they're going to help with movement of blood and lymph fluid. They're going to help with maintenance of posture and body position. Right now, if you're sitting up at a computer screen, or even if you're laying in bed, unless you're completely laying flat and not holding up any body parts, you have some sort of muscle stabilizing you somewhere. They will help stabilize joints, and they will help with heat generation. When your muscles use ATP, or energy, only 40% of the energy generated will be used for contraction of the muscle. The other 60% will be wasted as heat. So when you get cold, it's very easy for your body to warm up because it's very easy for your body to warm up because all it needs to have some skeletal muscle activity. Okay, so looking at skeletal muscle. Here we have a full muscle. We took one of these things right here, alright, which is a fascicle, and we blew it up. And then we took one of these things within the fascicle, which is a muscle cell, and we blew it up. And then we took one of these things from inside of that muscle cell, and we blew it up, which is a myofibril. And then inside that myofibril, these are thick and thin filaments. Myosin is the thick filament, and actin is the thin filament. So here we blew them up. So let's look at this chart here backwards. I have thick and thin filaments, actin, which is the blue stuff, and myosin, which is the purple stuff. I have a bunch of those that come together to form a myofibril. I have a bunch of myofibrils that come together to form a muscle fiber, which is a muscle cell. My muscle cell, or muscle fiber, is going to be wrapped up in what is called endomysium. Going back, when I take a bunch of muscle cells and put them together, they will form a fascicle, and my fascicle will be wrapped up in perimysium. And going back, when I take a bunch of muscle fascicles, those all together will form my whole muscle, and that will be wrapped up in epimysium. So let's look at these words. Epi is above or outside of, peri is around or within, and endo is within. So endo will be the deepest layer. Your epimysium will be dense, regular connective tissue, and it will also come together at the ends of the muscle to form a tendinous band that will be the attachment to your bones. It will be attached to your bones via Sharpie fibers. This attachment to the bones is so strong that before the attachment breaks, it's likely that that tendon can rip off a chunk of bone. Perimysium is going to be a fibrous connective tissue, so it's going to be a little bit thinner. It's not going to be as strong as this epimysium, but it'll do a good job of separating these fibers out. And then the endomysium kind of just separates everything a little more. When I think of this, I think of braiding hair, and my little girl is the one that made me think of it this way. So if I was to take each individual strand of hair and wrap it in saran wrap, that would be like my endomysium. So I'd take each cell and I would wrap it up in saran wrap. Now before I braid the hair, I'm going to take it and break it off into 3 separate pieces. Okay, and I will take each one of those separate pieces and once again wrap it up in saran wrap, and that would be my perimysium. So that would represent my fascicles. And then finally, for the outermost layer, the epimysium, I'd braid the hair. I have one solid unit, which would be my muscle, and I'd wrap it up again in a layer of saran wrap, and it would have its epimysium, its outermost layer. So, looking at some of our microscopic anatomy, we're going to be on this for a while, of skeletal muscle. Some of the muscle fibers are going to be short. Some of them are going to be longer. They will have diameter between 10 and 100 micrometers, and they can have a length of up to 30 centimeters. The smaller muscle fibers will be for delicate motions, and they won't be as strong. The bigger muscle fibers will be stronger, and they'll be for more blunt force. Each muscle cell will have multiple peripheral nuclei, so we call that multinucleated. Alright, they have lots of mitochondria. Why they need lots of energy? It makes sense. Our muscles use lots of energy to contract. They have glycosomes for glycogen storage. Why? One of the things our muscles will use to form energy is glycogen. They have myoglobin, which will be used for the storage of ATP. And they contain myofibrils. Your myofibrils will once again be the part of the muscle that will have the actin and myosin, and inside of those will be the actual contractile units. Wrapped around and deep within the muscle, you'll have a sarcoplasmic reticulum and T-tubules. T-tubules will propagate the impulse from the nerve deep within the muscle cell, and the sarcoplasmic reticulum will be used as a calcium storage, so it'll store calcium, and it will release calcium, which you'll see is very, very, very important in the whole contraction process. So our myofibrils, they're densely packed rod-like things. They take up most of the cell volume. They exhibit these things called striations, so we're going to talk about these on the next slide here. So the striations are caused here. Let's look at them. You see these right here? These are the striations. The striations are caused by this stuff here. So we have thick filaments, which is this dark red stuff. This is myosin. We have thin filaments, which is this blue stuff, which is actin. Wrapped around our actin, we have this stuff called troponin and tropomyosin. We'll see that in a little bit. Next, we're going to have this thing called the Z disc, and then we'll have the H zone. Okay? More things that we can see from this striation. We have an I band, which is also known as the light band, and we have an A band, which is also known as the dark band, and then we have an M line. The I band, the way muscle contraction works is this side and this side get pulled together. They both move. Let me grab a little pointer. They are both going to move in like that. Forward contraction. That shows up great, right? Red on red. Sorry. So they'll both move in forward contraction. Basically what happens is this little actin thing hooks in here and then pulls it that way. Actually, this little, myosin goes into the actin and pulls it that way. Okay, so here's a blown up thing. So our thick filaments run the length of the A band. We have our myosin. Alright, our thin filaments run the length of the I band, and that's our actin. These little myosin heads, they will attach to the actin and they will cock like a mouse trap and pull this in. Same thing here, they'll attach, cock like a mouse trap and pull that in. So blowing up a little bit larger, here's our myosin. Coming off of the myosin, there's a tail with a head, and the head will actually have two pieces. You see these two little pieces here? They walk like little legs. And this little dark spot on the tail. Okay, so these actin binding sites will want to meet up with these active sites on the actin. So we have this tropomyosin, which is the spaghetti looking stuff, and this troponin, which is this grape looking stuff. When calcium is released, troponin changes shape, pulls the tropomyosin out of the way, and now these are like magnets to one another. This will attach here and it will pull it together. We'll see that in the coming slides. So we have our sarcoplasmic reticulum. It will be the calcium storage, and we'll have the T-tubules, which invaginate deep into the myofibers, and that will help to propagate our impulse. So as that goes in, it tells this stuff to release calcium. Alright, here's our sliding filament theory. It's basically that the actin will move about the myosin. So if you go from here to here, the actin will move towards that H zone. Actin moves towards the H zone. The myosin stays put. The H zone will shorten or even disappear. So for the muscle to contract, we're going to need two things. Activation from a nerve at the neuromuscular junction, and excitation contraction coupling, ECC. The gist of that is that's going to be the little bit of time between the firing of the nerve and when the muscle actually contracts. So with the neuromuscular junction, here's what's going to happen. We have a nerve right here. Impulse travels down the nerve, and when the impulse travels down the nerve, you let calcium in to this thing right here. This is going to be the axon terminal. When calcium comes in, calcium causes us to release acetylcholine vesicles. Okay, so acetylcholine comes over here and is released. It diffuses across this right here, which is called the synaptic cleft. It meets up with these receptors here. When it meets up with those receptors, they open up this channel, and when that channel opens up, it has sodium flush in, potassium flush out. Okay, so that means that the muscle is now going to have the initiation of contraction, and that'll be when we get to the T-tubule. Also in here we have this stuff called acetylcholine esterase. It's right here, acetylcholine esterase. That stuff is going to be around to degenerate the acetylcholine. Why? So we stop muscle contraction, because as long as the acetylcholine's on here, we're going to have sodium flushing in, and that muscle is going to stay in a contracted state. We remove the acetylcholine, this channel closes up, no more sodium going in, my muscle can relax. So it's very, very important. So here's our sliding filament theory we'll talk a little bit more about. Our motor neuron reaches its action potential, we release the ACH, that's going to interact with our little protein channel. Alright, sodium moves into the cell, we release calcium from the sarcoplasmic reticulum. Calcium binds to troponin and tropomyosin. So here, let's see this. Here, I'll come back here. Calcium is going to bind to troponin and tropomyosin. We get our ATP driven power stroke, cross bridge detachment, and cocking of the myosin head. We're going to go through this right now. Okay, for the nerve to return to its resting state, you'll stop the stimulation. You will repolarize the muscle cell, so that means the muscle cell is going to like reset and get ready to contract again. Calcium is going to go back to the sarcoplasmic reticulum, and our tropomyosin blockade is reestablished. We talked about that for a moment earlier. And then the muscle fibers relax. So we talked of this excitation coupling contraction, or excitation contraction coupling. The gist of what it is it is a latent period. That is going to be from when the action potential reaches the nerve until this T-tubule can send it all the way down through all, this is the T-tubule here. The T-tubule goes deep into the muscle and gets all of the sarcoplasmic reticulum to release their calcium. Okay? Once calcium is released, we can start the whole muscle contraction process. So calcium gets released, it comes in contact with troponin, it changes its shape, and it moves tropomyosin out of the way. Now this binding site is available for my myosin head. My myosin head attaches to it, and it will walk and move it this way. Okay, so cross bridge formation, high energy myosin head attaches to the thin filament. You see that? So my myosin head will attach to the thin filament. When we detach the myosin head from the actin, we're going to use an ATP. It'll connect here and it'll detach. Alright, I think I have that in the next picture. Yeah, so the myosin head connects, it walks with its power stroke, and an ATP connects to it and it contracts. Next, we are basically going to use the ATP to recock that myosin head. So it's like using energy to reset a mousetrap. Okay, cocking of the myosin head, energy from hydrolysis of ATP cocks the myosin head. So the ATP attaches here, which breaks the bond between the actin and myosin, and then we use the ATP to reset that muscle, to reset the mousetrap for contraction. This whole process here will continue as long as we have calcium being released. So if we look back here, the reason why we get calcium release is the motor neuron fires, we release the acetylcholine, and sodium flushes into the cell. The T-tubules send the sodium in, and calcium gets released. So in order to stop this calcium release, we need to stop the release of ACH, or stop the nerve from firing. The way we stop the ACH is that acetylcholine esterase. Okay, so with the motor unit, we're going to have our motor unit, which is going to be a motor neuron and all of its muscle fibers it supplies. With small motor units, we'd find those in like your facial muscles in your hands, so those can do fine, small, delicate movements that do not require a lot of strength. With large motor units, you'll do those with stuff that requires a lot of activity, like your thighs, your hips, stuff like that. With muscle fibers from a motor unit, they won't be all grouped together in a fascicle. I think I have a picture here. They wouldn't all be grouped together in one area. They'll be spread out. The reason why they're spread out is when a fascicle contracts, it contracts 100%. It's all or nothing. The way I get a stronger contraction is I contract more motor units. So if I just want a weak contraction, I have a couple motor units fire. If I want a strong motor contraction, I can have all of my motor, or most of my motor units fire. But then what happens is, in order to sustain a contraction, they will like rotate in and out. Some will contract, some will relax. Some will contract, some will relax. And they'll be kind of switching off, helping out one another. Okay? Motor units and muscle usually contract asynchronously, and it will help prevent fatigue. That's what we were just talking about. Okay, so muscle twitch. Muscle twitch is going to be the smallest observable contraction that you can pick up in a lab, a myogram. So we're going to have three parts: the latent period, the contraction, and the relaxation. Latent period is our excitation-contraction coupling. So that's going to be the propagation of the action potential deep into the muscle. The contraction is going to be where the cross bridges are formed and the muscle actually shortens. And then the relaxation is the calcium going back in the sarcoplasmic reticulum. So our excitation coupling is this latent period here. Contraction is the building of tension, and then the relaxation is the pumping the calcium back into the muscle, sarcoplasmic reticulum. With your muscle contraction, all right, we need it. It can be graded, okay? The way we can change our muscle contraction is either by the amount of stimulation or the frequency of stimulation. So here, if we have a low stimulation frequency, it's like my muscle contracts, it's trying to relax, but then we get another stimulation, okay? And the muscle starts to contract harder, and then it relaxes. Oh, another stimulation. The muscle is starting to contract harder, and it's starting to relax. Oh no, another stimulation. So it keeps on getting stronger and stronger and stronger contraction because there's a low stimulation frequency. The high stimulation frequency, it would be like tetanus. Your muscle it's like a Charlie horse. Your muscle contracts and it just stays 100% contracted. All right. Next is our threshold stimulus. Threshold means when you meet threshold, you will have a muscle fiber fire. Like I said, for a weak contraction, you get a couple muscle fibers firing. For a strong contraction, you get lots of muscle fibers, or all of your muscle fibers firing. Once you meet threshold, right here, you get some muscle fibers firing. Higher strength of stimulus, more muscle fibers firing. Once you get to your maximum stimulus, all the muscle fibers fire. It doesn't matter if you even get more stimulation. Once all the muscle fibers are firing, they're firing. You're only so strong. You can only lift something so big. All right, so here would be small muscle fibers. They'd be a weak response. This would be fine motor movements. Medium muscle fibers, so they have a little bit stronger response than your large motor fibers. So these would be a strong response. This would be like your buttocks muscles, your gluteal muscles, stuff like that. Okay, with our contraction, there's going to be some different types. There's isometric and there's isotonic. Isometric means the muscle is staying the same length. So that would mean like I stuck my arm out and my arm is straight out to my side and you hand me a gallon of milk and I don't let my arm move. That's an isometric contraction. The length of the muscle is not changing. Isotonic means that the tonicity of the muscle is not changing. So I took that gallon of milk and I curled it. And I brought it down slowly. And I curled it and I brought it down slowly. So the amount of tension in my muscle is staying the same, but the length of it is changing. So iso is the same, metric is length. Iso is the same, tonic is tonicity or tension. With isotonic, we can have concentric contraction, where the muscle is shortening, or we can have eccentric contraction, where the muscle is lengthening. Okay, so the way my muscles are going to be getting energy is going to be three ways. There's direct phosphorylation, where I will use creatine phosphate, and it will combine with ADP to form ATP. ATP is going to be our main energy source for everything in our body. Okay, creatine phosphate turns into creatine, and then we're just left with ATP. We can undergo glycolysis. So this is our anaerobic pathway. In glycolysis, it's literally the breaking of glucose. You break apart glucose and you make some ATP. Or we have our aerobic pathway. All right, that is going to make 32 ATP per glucose. It will make lots and lots of energy. All right, so anaerobic, glycolysis, we use no oxygen. It happens in the cytoplasm. We make just a little bit of energy. We'll use this for quick, short contractions. Aerobic, this is going to be when you're making lots of energy and you're at moderate exercise, like aerobic exercise. Okay, we'll make lots and lots of energy, and this will give you sustained exercise abilities. Muscle fatigue. The true definition of muscle fatigue is when the muscle is unable to contract. The reason for it, ionic balances, so you're screwed up with your potassium or calcium. That's going to interfere with that excitation, coupling, contraction. If you have lactic acid buildup, that's going to drop the pH, and that's going to make it so you can't do your anaerobic production, your glycolysis. If you're exercising too long, you damage the sarcoplasmic reticulum. This is actually why you get sore the next day. You exercise too long, the pH drops, we get some sarcoplasmic reticulum damage, and your muscle's sore. So when the muscle rebuilds, it rebuilds stronger and more efficient for the next time. And then if you get total lack of ATP, this is rare, but if it occurs, it would occur from something like tetanus. The muscle's in constant state of contraction for a long time. Okay, next ones are oxygen deficit. So you need extra oxygen after exercise to do a couple things. Replenish your oxygen reserves, restore glycogen, and return your ADP to ATP and make your creatine phosphate. All right. It's also going to help with the conversion of lactic acid to pyruvic acid. All right, we spoke of this earlier. When your muscles are contracting, only 40% of the energy released in the muscle is used for contraction. The rest of the 60% is given off as heat. All right, when we have our muscle contraction force, it can be determined by a couple things. The number of muscle fibers, the size of the fibers, the size of stimulation, and the length-tension relationship. So bigger muscle fibers contract harder than smaller muscle fibers. Larger groups of muscle fibers will contract stronger than smaller groups of muscle fibers. And if you have a higher stimulation, just as long as you are past your threshold, you will have more muscle fibers fire, versus if you have a lower stimulation, you'll have less muscle fibers fire. With your length-tension relationship, the gist of it is when you're in the mid-zone of your sarcomere length, it's the strongest. All right. When your muscles are overstretched, they're weak, and when they're at the very end range of motion, they're also a little bit weaker. That's the reason why when you're at the gym, you see these people working out and they're just moving the weight just like a couple inches. They're not going through the whole range of motion. The reason why, they feel stronger. They really can't lift. They can't move the weight throughout the whole range of motion. So they use a lighter weight or a heavier weight to move it less distance. Okay, with our duration and velocity of muscle fiber, okay, we're going to talk about these guys now. We'll have our speed of contraction. It can be slow or fast. So we'll have slow fibers or we'll have fast fibers. And then the other thing we can attach to it is oxidative or glycolytic. Oxidative fibers use aerobic activity. Glycolytic will use our anaerobic activity, which is glycolysis. Moving to the next slide because this does a better job of describing them. So we have our three fiber types: slow oxidative, fast oxidative, and fast glycolytic. Slow oxidative fibers are good for postural stuff, and these will be good for like running a marathon or something like that. So these muscles will contract slow. They don't need a lot of ATPase. They use aerobics because they're going to use a lot of oxygen. They have lots of myoglobin because myoglobin acts to store oxygen inside the muscle cells. They don't need a lot of glycogen storage because they're doing the aerobic pathway. They will be the first muscle fibers to fire, and they will be slow to get tired. This is going to be a nice red steak. So cows are red meat because they have slow oxidative fibers. The meat is red because they need endurance stuff just to kind of stand there and move slowly. Fast oxidative would be like a sprinter. You need like a minute or two or three of activity. These muscles will be fast contraction. All right, they will do some aerobic pathways, some anaerobic. They'll still have a lot of myoglobin. All right, their glycogen stores will be in the middle. These will be the second muscles to contract. All right, and they will be intermediate. So this will be kind of in the middle. These would be like sprinting or walking. And these will be red to pink. This will be like a little bit lighter color. Fiber diameter will be intermediate, and they'll have lots of mitochondria. Fast glycolytic. These would be like a defensive lineman or an offensive lineman in football, or somebody who just bench presses all the time. These muscles will contract quickly. They only need a short burst of energy, so they have the myosin ATPase. They do this aerobic pathway, which means it's a quick contraction. They have low myoglobin concentration. Why? They don't need oxygen. They have high glycogen. Why? Because they need that glucose to be able to contract quickly and just for a short period of time, and they fatigue quickly. So this will be short, intense, powerful movements. All right, these are large, white, pale fibers. Okay, so aerobic exercise. If we do aerobic exercise, it's going to lead us to grow more aerobic or slow oxidative and fast oxidative muscle fibers. So what happens here, we get increased muscle capillaries, more mitochondria, more myoglobin, and we'll have greater endurance and strength and resistance to fatigue. If you do anaerobic exercises like just straight weightlifting at low reps, your muscles grow thicker, they have thicker muscle fiber diameters, and the reason for that is because those thicker muscle fiber diameters are stronger. They can have a blunt force quicker. So these will have more mitochondria, all right, the myofilaments and the glycogen stored will go up. So basically look at this past chart here. Whatever you need to increase, everything on this chart would increase if you do endurance exercise. Everything on this chart on this side would increase if you do weightlifting type exercises. So that's why a weightlifter would have big, strong, bulky muscles, and a marathon runner would have slender, lean, long muscles. Okay, next we're on to our smooth muscle. So this is going to be found in the walls of our hollow organs except for the heart, except for the muscle wall of the heart. That is our myocardium. These will usually be found in two layers, a longitudinal and a circular layer, and they will be responsible for peristalsis. You have alternating contractions or relaxations of smooth muscle layers that mix and squeeze substances through the lumen. Okay, so you get a longitudinal layer that will contract, it'll shorten. Okay, and then you get a circular layer that contracts to hold that in place. Then the longitudinal layer relaxes, and it keeps the circular layer stays contracted, and it keeps whatever moved down. Once again, longitudinal layer contracts, it moves it down the way. Circular layer contracts, holds it there. Longitudinal layer relaxes. All right, so it's kind of like climbing a rope. So here we go. And if you look at this smooth muscle, it doesn't look like skeletal muscle. It's not striated. Here's our circular layers, and the longitudinal layers run lengthwise. All right, so these are spindles is how they're described. All right, so they're spindle shaped. All right, they only have an endomysium, and they have a less developed sarcoplasmic reticulum. They do not have myofibrils or T tubules. And they're, instead of a neuromuscular junction, they have these things called varicosities. I have it in a couple slides from now. So here's our skeletal muscle. It's nice and striated, and it's a nice, easy to identify pattern. Here's our cardiac muscle. It looks similar to skeletal muscle, but it looks like somebody took it and pulled it apart. We only find cardiac muscle in the walls of the heart, in the myocardium of the heart. And then here's our smooth muscle. It looks like a spindle. Okay, skeletal muscle, it'll have its endomysium, perimysium, and epimysium. Cardiac muscle and smooth muscle will only have the endomysium. Skeletal muscle will have its T-tubule. Cardiac muscle will have its Z-disc, which is similar. Skeletal and cardiac muscle both have the sarcoplasmic reticulum, while the smooth muscle doesn't. Here's those little varicosities. So there's these like fluid-filled sacs that are placed all over the smooth muscle. So the smooth muscle kind of contracts in this rhythmic like pattern. Okay, with smooth muscle, it's going to have 1 thin filament to 13 thick filaments. So that means thin filaments are weak, thick filaments are strong. So it's going to be a slow, endurance, repetitive motion. With your skeletal muscle, you get 1 thin filament to 2 thick filaments. So that means that they're going to be able to have a more unified, more strong, boom, done contraction. You get no troponin. All right. There's no protein calmodulin. Just know there's no Don't worry about exactly what it is. Just know there's no protein calmodulin. And then their myofilaments are going to be spirally arranged, hence the spindle shape. So it's going to cause a corkscrew type of contraction rather than just like a shortening and elongation. Shortening and elongation. So that's this part. They'll be spirally arranged because of the spindle shape, and it'll cause a corkscrew contraction. Okay, with smooth muscle, it will contract slowly, The muscles are going to be electrically coupled by gap junctions, so it will have a nice rhythmic contraction, and some of them will be self-excitatory. When I think of this, I'm thinking of the heart. Alright, with regulation of contraction, if we have increased neurotransmitters, that means increased contraction. That's the gist of it. Increase acetylcholine in our skeletal muscle means increased contraction. Same thing here. Alright, special things with smooth muscle. Stretch relaxation response. So when smooth muscle stretches out, it has a response to relax for a second. It can relax until it is stimulated and told to, hey, it's time to contract. Another thing is that smooth muscle can stretch out quite a bit. Your stomach is about the size of your fist. Think about how much food you've eaten at some points in your life and some days in your life. Sometimes you eat this huge plate of spaghetti that's 3 times the size of your fist. Somehow it fits into your stomach. Why? It can contract down to its normal, way below its normal resting length, and it can just relax to a huge amount. Then hyperplasia. Smooth muscle cells can divide. The only place the most specific example where it happens would be in the uterus. For females in puberty, it is going to grow so it can hold a baby, and then once they get pregnant, it will once again, that uterus is going to have to grow as the baby grows so it can sustain it, and it can be strong enough to push the baby out. With our smooth muscle, there's going to be 2 types: single unit visceral and multi-unit. Single unit visceral will contract rhythmically, okay? It'll have spontaneous contractions, and we'll have these opposing sheets. With our multi-unit, these are going to be the way it'll work with our large airways and our arteries. So it'll be like vasoconstriction, dilation. It'll be just like blocking off an area or opening up an area. And that's it.